Today we'll be discussing the first prompt for the 5th genetics mini video. We are genetic counselors, and today we'll be seeing one of our clients named Susie. Susie has come to us to let us examine her DNA to help us better understand the process of transcription and translation, as well as point mutations. On this first slide, we see Susie's DNA. We have the coding strand on the top shown in the 5' to 3' direction. Our first question is what happens when transcription and translation occur? This slide portrays the process of transcription. Here we see the DNA from the first slide, and we can see the coding strand being used to produce the RNA strand below it that is pictured in the 5' to 3' direction. We matched up the base pairs. For example, the G in the coding strand is now a C, and the T is now an A, and the A's are now U's, and the C's are now G's in the RNA sequence. After transcription, translation occurs. You can see here that the transcribed RNA sequence is divided into groups of 3 anticodons. The anticodons then pair with the codons to perform translation. In Susie's genetic material, you can see that we found the AUG start codon coding for MET, and then continued coding until we got to the stop codon, which in this case was the UGA anticodon. Our codon sequence for Susie was MET, TRP, ILE, THR, and the stop codon. On this slide, we are now going to talk about some of the problems that we see in Susie's DNA. On this slide, we see the coding strand and the transcribed strand. After writing out the codons that match the anticodons seen in the transcribed strand, we find that a point mutation has occurred. We see that a transition from A to G has occurred, causing the ILE codon to turn into a VAL codon. This is a missense point mutation because the change in base pair now codes for a different codon. On this slide, we have another problem with Susie's genetic material. We see the coding strand still on the top and another transcribed strand below it. On this slide, we had a transition occur, changing a G to an A, which causes a nonsense mutation. The A being placed in this sequence caused the anticodon to now code for stop codon, which causes the translation to stop. Here we have another example of a point mutation in Susie's genetic material. We see here we have a transition occurring, changing the base pair from a T to a C. This causes a silent point mutation. A silent point mutation means that even though there was a mutation, there was no difference in the codon strand that came out of it. In this last example of a point mutation in Susie's genetic material, we see that the A changed to a C in the coding strand. This is a transversion and creates a read-through mutation. This is the case because with the change, the codon would typically code for stop, now codes for cis, so that strand continues to be translated past where the codon would be. These are the references that were used for this presentation.